Today we're going to be talking about point mutations, and as you can see here, I have a strand of DNA and a strand of mRNA. The strand of DNA is the coding strand that I have transcribed into the mRNA sequence. I'm going to be using Fruit Loops to kind of color code and denote each nucleotide. Here we have our amino acids that were derived from the mRNA codons, which are methionine, which is the start codon, tryptophan, asparagine, alanine, cytosine, and the stop codon. And this is our normal sequence. So I'm going to make some mutations to show the different types of point mutations we have. I did the DNA, but I'm just going to go ahead and show you my mutated RNA because it's easier for me to explain it since we're going to be talking about the amino acids. And to show you the difference between the DNA sequence and the mRNA sequence. So the DNA has thymine and the mRNA has uracil instead. So I have switched out all the thymines in the DNA sequence to uracil in the mRNA sequence. Also, the coding strand and the mRNA sequence are both opposite and anti-parallel to the template strand, which is why they both go from 5' to 3'. Single base mutation that results in the change of a previous amino acids to a different new amino acid. So here, what I have done, and I have indicated it with this, is that my UGG has changed to an AGG, which codes for a different amino acid. So the blue Fruit Loop has changed to a red Fruit Loop, and then causing the codon to change to a new amino acid. So instead of tryptophan, it's now arginine. And this is called the Missense mutation, like I said earlier. Here I have denoted the nonsense mutation. And a nonsense mutation is when a single base mutation results in the change of amino acid to a stop codon. And here we can see that UGC has been mutated to UGA, and UGA is the stop codon. So it went from cytosine to stop. Here I have denoted the silent mutation, which is a mutation of a single nucleotide that still codes for the same amino acid even after mutation. So here I went from a cytosine to a uracil, and I still have the same amino acid, which is asparagine. This from cytosine to uracil, this mutation is also called a transition mutation since they are both pyrimidines. And for the Missense mutation, I had a transversion mutation because it went from a purine to a pyrimidine. It went from a U to an A. For the nonsense mutation, I also had a transversion because it went from a C to an A. So it went from a pyrimidine to a purine. Moving on, here I have denoted the last point mutation, which is the read-through mutation. And the read-through mutation is when a single nucleotide is mutated and it results in the change of a stop codon to an amino acid. So here we have UGA, which represents a stop codon, that's now being mutated to UCA, which is serine. And serine is an amino acid, so we went from a stop codon to an amino acid, which makes it a read-through. So since it doesn't stop, it just keeps reading, and it reads proline and glycine. These are all going from 5' to 3' since they're mRNA sequences. And I hope this gives you an idea on point mutations and their effect on the translated amino acid sequence.